The E. coli species is composed of many thousands of variant strains, some of which are pathogenic. In this study by Sansonetti and coworkers, they investigate what is the minimal set of genes needed to convert laboratory K12 strains into a strain functionally equivalent to Shigella flexneri. Shigella is E. coli's closest cousin on the phylogenetic tree, but from a clinical perspective, the two organisms are quite different. While most strains of E. coli are harmless commensal bacteria, Shigella are usually pathogenic, causing a particularly nasty form of dysentery. Shigella infects animals by oral ingestion and enters the intestines. Like many other pathogenic enterobacteria, Shigella invades M cells and then macrophages. Shigella then bursts from the phagocytic vacuole and is able to spread from epithelial cell to epithelial cell by polymerizing actin from one of its poles. In this way, it is able to jet around inside the cytoplasm of the cell and propel itself across cell junctions. This video is actually of listeria, but a similar behavior of actin polymerization-based locomotion is observed when an epithelial cell is invaded by Shigella. The experiment they do in this study involves transferring regions of the Shigella genome into E. coli K12 and then performing assays to determine what aspects of the pathogenic mechanism transfer. Experiments like this were very popular in the 1980s when much of what we know about the molecular basis of pathogenesis was unraveled. In this study, they want to define what is the minimal set of virulence factors needed to make Shigella phenotype. And more globally, they are getting at the question, what makes something a pathogen? They reconstitute Shigella in K12 piece by piece, starting with the virulence plasmid. Shigella encodes a 220 kilobase episomal DNA that is known to contain many of the virulence factors, including its type 3 secretion systems and the genes for cell-to-cell spreading. They put this plasmid into E. coli and then start adding genomic loci from Shigella. Each variant strain is subjected to a battery of assays that each probe different aspects of the pathogenic mechanism. They start with strain 395-I of E. coli, which is a K12 Mg1655 lineage strain, and they transfer in the virulence plasmid. At 220 kilobases, transformation using conventional methods is inefficient, so usually such large DNAs are mobilized by conjugation methods instead of heat shocking or electroporation. The resulting strain is able to invade HeLa cells and escape the vacuole, but all the other tests they perform fail. They then start putting in Shigella genome loci to see what else is required. At the time this study was performed, the genome of E. coli had not been sequenced and none of the tools like gene synthesis or PCR existed. Thus, they performed their study using more traditional genetic mating experiments using conjugation. Conjugation is a process in which DNA is transferred from one DNA to another starting at specific loci. By controlling how long the bacteria are able to mate, you can control how much of the genome gets transferred. The success of a mating experiment is known by the transfer of genes adjacent to the ones of interest. By starting with a histidine auxotroph, mating can result in repair of the growth defect and thus be selected. Thus, any virulence factors that lie near the His locus can be transferred and selected based on this phenotype switch. They show that transfer of the His locus leads to successful production of O antigens. Group-specific antigen 3.4 or 3,4 somatic antigen refer to Shigella flexneri's O antigen, and it's now known to be encoded near the His locus in Shigella. O antigens are primarily to avoid phage infection and opsonization, but also they can be necessary for the proper expression of other virulence factors. Experimentally, there is a simple phage electrophoresis-based assay for detecting O antigens and quantifying the degree of polymerization. Another locus that gives rise to phenotypes in their assays is virulence factors near the pure E locus. Restoration of this locus results in bacteria that pass their test for cell-to-cell spreading within a HeLa monolayer. They refer to this virulence factor as KCP, but later studies revealed that the gene conferring the cell-to-cell spreading was OMP-T, a surface-expressed protease needed for functional expression of VIR-G, which is the protein required for actin polymerization-based locomotion. Another important locus they found was associated with the ARG-MTL region of the chromosome, and addition of this locus enables the bacteria to pass the rabbit ileal loop test. This assay quantifies fluid production, which is a surrogate for extreme diarrheal effects observed during Shigella infection. This region of the genome was later shown to encode Shiga toxin, which is confirmed to be necessary and sufficient for this phenotype. The actual data of this study is a series of genetic compositions and what they do with respect to 4 different assays related to virulence. Using mating-based genetics, they show that you need to add the type 3 secretion system and effectors from the virulence plasmid, the O antigen, OMP-T, and Shiga toxin to pass all the standard pathogenicity tests for Shigella. One question this study raises is whether this set of genes is truly sufficient for virulence. The claim of sufficiency is always problematic in biology because it is challenging to demonstrate that some other unknown factor is not contributing to the observed phenotype. Since E. coli and Shigella are so similar, it is entirely possible that they share genes that are also required for the observed phenotypes. However, this sort of transfer study allows very strong claims about what is required for a phenotype. Another point to make about this study is that such things are rarely done anymore, in large part due to biosafety concerns about potentially engineering pathogens in the lab. In fact, any experiment involving the transfer of virulence factors receives specific scrutiny under the NIH guidelines, which regulate research in academia and indirectly in industry. Such experiments are considered potentially hazardous and require special review.